All right, good evening and welcome back, everyone. I hope you're having a beautiful Tuesday. Let's continue our conversation about genetics. Today, we're going to tackle what I think is for some people the hardest part of the class, which is operon logic. That is to say, how bacterial genes are often regulated. So let's get to it. The first thing I want to do is just a little warm up. I'm going to cold call a couple of people and then open it up for raise your hand. So again, as you know, my policy is that if you're not sure of the answer and you don't feel comfortable guessing, you can always pass. But let's start with this term. How would you define a constitutive gene? Let's begin with Surmeet.
Those are the genes. Those have codes for the proteins we need all the time. For example, they're made constantly. For example, ribosome we always need, like RNA protein.
Great, that's a good definition. Yes, constitutive gene is one that is not regulated. It's just always being transcribed. The gene is in the genome. The cell is always making mRNA and then expressing proteins. That's a constitutive gene. Another example that's not a protein coding gene would be like a tRNA or a ribosomal RNA, where that gene is constantly being transcribed because we need it all the time. What about a repressible gene? Let's go to Haley. Repressible. Do you have a definition for repressible? And if you want to pass, you can always pass.
Those are the genes that are normally on unless the environment turns them off.
Exactly right. Good answer. So these are genes that normally we would need. We would have them on sort of as a default, but occasionally some environmental condition makes it make more sense for us to not turn them on. And so we turn them off. Repressible. And last but not least, inducible. Tao, would you like to take a stab at this one? That's no problem. Let's next try Megan. Do you have an answer for this or a definition you'd like to share?
I'll pass. Sorry, I just got home.
No problem. John, what would you like to say about inducible genes? We've had ones that are always made. We have ones that are on by default, but the environment can turn them off. John, do you have an inducible gene definition? Usually they're off because we usually wouldn't need them or we don't want them on by default, but the environment can turn them on. Exactly. Good job, you guys. Just to remind you that in bacteria, it's very common for genes to be organized into operons, where a good example would be the enzymes that are all part of the same metabolic pathway might be co-regulated by the same promoter. This is where RNA polymerase is going to sit down. And these genes in operons are often regulated. And if they are regulated, there is an area between the promoter where RNA polymerase binds and the genes, which it is going to transcribe, called the operator. And what binds at the operator? Lori, do you know?
I'm going to pass.
Not a problem. Nikki, how about you? What protein binds to the operator to help regulate the expression of these genes? I see. Okay, yeah, Nikki's going to pass as well. Let's see. Sarah, what would you say? What binds the operator?
I made that diagram and I wrote control sequence, but I don't know if that's right.
Well, you're on the right track because the operator is definitely a control sequence, right? It is. And there's a protein that binds it in order to prevent transcription when that's appropriate. And so we're looking for the name of that protein. Tiffany, do you know what it is? All right, let me just open it up for a raise your hand. Anybody who knows what it is, go ahead and raise your hand and tell us what it is. Amy, go ahead.
Would that be DNA?
It would not be DNA. This is
Oh.
already DNA, but that's a great guess. So it is certainly part of the picture, right? This is the DNA. What we're looking for is what's going to bind this region of the DNA called the operator. Let's see. Oh, we now have some answers in the chat. Excellent. Tiffany and Caddy say, is it the repressor? They are correct. All right, good job, you guys. The repressor is a special protein that binds this sequence in order to prevent transcription. And in these situations, we have to remember how repressors work. They're not like they bind and then they just sit there forever. They can come off and allow transcription to proceed, and they're regulated or affected by some environmental signal. The repressor itself is a gene. It has its own promoter. It is always expressed. Since it is always expressed, this type of gene is constitutive, repressible, or inducible, guys. What do you think? Put that into the chat. The repressor itself is always expressed. Is that constitutive, inducible, or repressible? This gene, the repressor gene. What do you think? Great. Most of you put constitutive, and you are correct because it is a gene that's always expressed, and that's the definition of a constitutive gene. Okay, let's have a closer look at how repressors work. So here is an example where the gene that is the repressor, constitutively expressed, meaning it's always being made into message, and that's always being translated. And here it is binding to this sequence that is in between the promoter and the genes. And again, what is this sequence called? This control sequence has a name. Where the repressor binds is called the what? Put that into the chat. Exactly right. That is the operator, okay? So if the repressor is bound to the operator, we do not get transcription. If it goes away, that allows transcription. Okay, now if this was the only thing that happened, then life would be really boring and really wouldn't happen, because if repressors, all they did was just sit on operators, and since they're constitutively expressed, they'd always be there. We would never get transcription, nobody would make protein, life would not exist. So clearly that's not the whole story. Repressors are allosteric, which means that they can change their shape depending on whether something else binds to them. They have two shapes, okay? So here are the two shapes of a repressor. One that functions and binds the operator, and one that doesn't function and doesn't bind the operator. And which shape they are in depends on whether they are bound to their allosteric regulator. And that can be many different things depending on the repressor. This is just one example, okay, of one type of repressor. And this is only some repressors. When they are first translated, they're in the non-functional state, which means they're not here. And so transcription is happening by default. What type of genes do we want to be regulated with this kind of normally non-functional repressor? Genes that are repressible or genes that are inducible? Put that into the chat. We have this type of repressor that's normally not functional. It has to actually be changed in order to do its job. So do we want this type of repressor that would normally allow a default transcription for a repressible or inducible gene? Let's remember how those genes work. Repressible ones are normally being transcribed by default and can be shut off by some environmental cue. Inducible genes are normally not transcribed and can be turned on by some environmental cue. So what do we have here? If our repressor is born in the non-functional state and it's not here and transcription is happening, then transcription is happening by default. So is that the definition of repressible or inducible? So it's a 50-50 split in the class. It's understandable if you didn't get this right, because this is like, there's a lot of double negatives in this logic circuit. But this is the kind of repressor that controls a repressible operon. Because since it is not normally doing its job, then the default state for transcription is to be on. And repressible genes are regulated like that. Their default state is to be being transcribed. Some environmental signal can come along, bind the repressor, make it function, and that will stop transcription. But it's an environmental signal, not the default situation. And this environmental signal, whatever it is the co-repressor. Okay, so just a reminder, a repressible operon is one that contains genes that are usually on or off. Repressible means that the genes are normally transcribed or not. Usually transcribed or not, what do you think? Repressible means they're usually on, that's right. And therefore, and something in the environment can turn them off. An example is catabolic enzymes, such as the ones that make amino acids. And the operon that we will use as our example is the tryptophan operon. That is the one that has the genes to make the enzymes to synthesize tryptophan. Inducible operons contain genes that are usually catabolic. They usually break something down. They're usually on or off, usually transcribed or not transcribed. That's right, they're usually not transcribed. It's something in the environment has to turn them on. This makes sense because we don't want these genes to be made that metabolize carbon sources unless the carbon source is actually present. Let's see if we understand that well enough to see, to define this picture. So in this example, we have a, let me just go back to this picture. I want to point something out. Okay, here we go. Just to sort of say this again more clearly, there are all repressors have a shape that binds the operator. Some repressors are made and immediately assume a functional shape, and they immediately bind the operator, and they by default stop transcription. And if we want to remove them, we need some kind of environmental signal that binds and changes this to a non-functional shape. Other repressors are born in the non-functional shape already, and their default is to allow transcription. And an environmental cue can change their shape. So now they sit in the operator and stop transcription. So is it clear that there's two types of repressors, and that is the consequence of that is that you either have a repressible or inducible operon? Is that clear? Yes, no, or kinda? Surmeet, you have a question.
I understand the first one. The second one, active repressor protein that needs something to bind on that change the shape and stop the transcription. What is that type?
So there isn't a specific, they're both repressors, and it's more that you want to think about which type of repressor you want in each circumstance. So there isn't a formal name for them really. You just have to know that all repressors have the ability to bind the operator, but only some of them are born in that shape. Some of them are born ready to bind the operator, already with no help. They just fold into this shape straight away. And others have to be coaxed into it when an environmental signal shows up. Does that help? Okay. Okay. So let's have a look at this example and see if we can decide whether this is describing an operon that is repressible or inducible. So in this situation, what you can see is that the repressor, which remember is constitutively expressed, so it's always around, when it's made, it immediately folds into the correct shape to bind the operator. And as a consequence, the default situation is that there is no transcription. Here's the RNA polymerase, here are the genes, and there's no transcription because this repressor is already born ready to bind the operator. It doesn't need help, it's already the right shape, and it's by default stopping transcription. If you want to remove this, this requires some kind of environmental signal that will bind to and relax the repressor so that it leaves the operator, allowing transcription. Is this the definition of a repressible or inducible gene? The default state is that transcription is not happening, and we need some kind of environmental signal to allow transcription to happen. And notice what that environmental signal is called. Nikki is right, John is right. Yes, Haley. Okay. Remember that inducible genes are ones that are normally not transcribed. That's their default state. And it's some environmental signal that causes them to be expressed. This is common for things that are enzymes you need to break something down. You don't really want to make this unless whatever its substrate is actually present in the environment. Okay, let's look at another situation. This is an example where the constitutively expressed repressor, when it folds, it is not ready. It is lazy. It is born lazy. It's not in the right shape to bind the operator. So the default state of transcription is what? The default state of transcription is that it is on, right? If we want to turn this off, if we want to regulate this and turn it off, some kind of environmental cue has to come along and bind the repressor and talk it into, coax it into doing its job and sitting in the operator. When that happens, transcription stops. The default situation is that transcription is happening. The environmental cue can turn transcription off. Is this repressible or inducible? Correct. And that is the definition of a repressible gene. So that's how the tryptophan operon is regulated. And we are going to use it as our poster child for other operons just like this. It is an anabolic operon, meaning that it has codes for enzymes that build stuff. In this case, the enzymes that make the amino acid tryptophan, and there are 3. Glucose is the starting material for tryptophan in E. coli. Note that making tryptophan is not inexpensive. It is not free. It costs something because we're making something. So energy is required to do this. But we need to always have tryptophan pools in our cells. Because tryptophan is an amino acid, we need all the amino acids to be available in case we need to make a protein with tryptophan in it. We never know what our next protein that we might need to make is going to be. So what kind of circumstance might cause us to stop making tryptophan? Well, what if tryptophan was suddenly plentiful in the environment? Does it make sense to continue to spend energy creating the enzymes that make more tryptophan? Or do you want transcription of these to temporarily halt? Yeah. If there's suddenly a whole bunch of tryptophan in the environment, it doesn't make any sense to continue spending energy making more. Why not use up the environmental stores? And then when you run out, you can go back to making your own. So again, let's think about what the default situation is. The default, what we want is for these enzymes to be made continuously as a default state. But if tryptophan, the environmental cue, is suddenly elevated, we want to turn transcription off. Is this a repressible or inducible operon? Remember what the definition of these terms is. Repressible means usually transcribed, can be turned off by an environmental signal. And that's what's happening here. The default state is we're making these enzymes so they can make tryptophan. And then the environmental cue of high tryptophan would cause this to stop. This is repressible. Now, if we want to achieve this type of regulation, if what we want is the default to be transcription is happening, do we want our repressor to be sitting in the operator all the time? That's right, we don't. So if we have two options, you could pick a repressor that is born lazy and doesn't bind the operator so that transcription can happen by default. Or you could pick a repressor that's born ready, always binds the operator. Which one are you going to choose, lazy or ready? Let's think about what will happen in each case. Let's choose, the first suggestion in the chat was ready. So let's choose the born ready repressor. We choose the born ready repressor. That means it's always in the operator. Transcription is off. The default is that transcription is off. Is that what we wanted? Yeah, we want the lazy one, right? We want the repressor that is born and is kind of like, you know what, things seem fine. I don't need to intervene. Go ahead, transcribe away. What should be the environmental signal? What should be the environmental signal that causes the repressor to get its act together and start doing its job and stop transcription? What should that environmental signal be in this case? Yes, Haley. We should. Yeah, that's what Haley said. Yeah, high tryptophan should be our environmental cue. It should go talk to the repressor and say, yo, you remember how you have this job to do? Well, it's time. You know, you got to move out of the house, dude. You can't just stay and play video games on the couch and eat potato chips. You got to go get a job. So that is a repressible situation. What makes it hard to remember is that whatever the repressor is doing is the opposite of what transcription is doing, right? If we want transcription to happen, we have to have a lazy repressor. If we want transcription to stop, we have to have a functional repressor. Okay, so here again in pictures now, we've got the tryptophan operon. Here are the genes required to make the enzymes to synthesize tryptophan. And here's the promoter, here's the operator, here is the repressor. Is it born ready or born lazy? Yeah, it's born lazy. It's not doing its job. It's not sitting in the operator, but that's good news. So transcription can just happen. That's the default state. It's happening, it's happening, it's happening. What is the environmental cue that we need it to cause this repressor to change its shape and bind the operator and stop transcribing? What is that environmental cue that should be what this repressor cares about? Tryptophan, right? There it is. This is tryptophan. When tryptophan binds to this, it turns it into a functional repressor. It gives it that motivational speech needed to get its boots on the ground, and it goes and sits in the operator. And what happens to transcription? It stops. Hallelujah. It stops. That's what we wanted, right? Because what is the job of these genes when they're translated is to make more tryptophan. Now we have lots of tryptophan, so now we can stop making more. Okay. All right, so let's go over and see if we can make sense of this. When tryptophan is absent, there's not tryptophan in the environment, the repressor is functional or non-functional? Right, the repressor is non-functional if there's no tryptophan. And as a consequence of that, transcription is happening or not happening? It's happening. Good. That's what we wanted. Now, let's add some tryptophan to the environment. It goes and talks to the repressor, and now the repressor is active. And it goes and sits on the operator, and then what happens to transcription is it's off, it stops. Does that make sense? Yes, no, or kind of? Everybody weigh in here. It is a double negative tau. You're absolutely right. It is. And you know what I'm going to tell you? I'm doing this in words as well to sort of help you get to the point where you're comfortable with us on a test. But a picture is worth 1,000 words. You should draw pictures. You need to have those pictures. In your study guide, there should be a picture. When you go take the test, you need to bring a picture. Okay, so again, the tryptophan operon is one that we want transcribed by default. We want tryptophan, the environmental cue, to turn it off. So we have to have a repressor that's born lazy to allow that to happen. And then when tryptophan is high in the environment, we need it to come and talk sense into that repressor and get transcription to halt. Now, this is not a forever deal, okay? When something binds something else in living systems, it's typically a transient bond. So this tryptophan is bonded here, but occasionally it's going to go and have a potty break or whatever. And for a second, this repressor is going to revert to this situation. As long as there's lots of tryptophan in the environment, like more than just this one molecule, it's all over the place, then this one goes for a potty break, another one steps in and fills its place, and the repressor never gets to take a break. But as soon as we start using up all that tryptophan, and now we're really low, this goes to take a break, and suddenly there's no more tryptophan in the environment, and we have reverted to this situation, and we can get transcription again. Now, of course, that's not how everything works. Let's compare that to the lactose operon. The tryptophan operon is anabolic. It costs energy. It makes enzymes for something that we need most of the time, and we want to just turn off some of the time. The lactose operon encodes genes that are enzymes to break stuff down. Do we need these on by default? I will, Sarah, yes. The lactose operon encodes enzymes to break down lactose. do we always want these enzymes to be made? Like, should the default state be, no matter what's going on in the environment, let's just make sure we have lactose breakdown enzymes? The answer is no. And the way to think about this is it costs energy to make these enzymes. And what if there's no lactose in the environment? Does it make sense to be making enzymes for stuff that isn't even around? And if that's the case, if we don't want to make these by default, if the default state actually should be that we're not making these enzymes, what should be the environmental cue to have their transcription come on? Yeah, the presence of lactose in the environment should be what triggers the transcription of these genes. Now, it turns out not to be as simple as that, just to give you that sort of spoiler alert, but nonetheless, we're going to start with that, okay? If that's the case, if what we want is by default no transcription, and then the environment has to go in and turn things on, is that the definition of an inducible or repressible operon? Yeah, that's inducible. That's textbook inducible. Okay, so we only want these on when lactose is present. The default state should be no transcription. Given that that's the case, what kind of repressor do we want? Do we want a repressor that's born ready, sits in the operator, and by default stops transcription? Or do we want a repressor that's born lazy and just lets transcription happen willy-nilly? And my hint to you is that it's going to be the opposite of what we just talked about. So think about what you thought the tryptophan repressor should be. And then it's going to be the opposite of that. Yeah, we want the repressor to be born ready and active and doing its job and sitting in the operator and stopping transcription. Remember, whatever is happening with transcription is the opposite of what's happening with the repressor. The repressor is active, transcription is not. We want our default state to be no transcription, so we want our repressor to be doing its job by default. Does that make sense? Fabulous. Good. So let's see how this works. You can see in this picture that this is an example of the type of repressor that's born ready, born active, born functional. No sooner is it translated than it is already in the right shape with no further help to sit in the operator. And consequently, what will happen with transcription? The default is that transcription will be on or off. Remember that as long as the repressor is in the way, transcription cannot happen. Right. If the repressor is here, it's repressing transcription. So that's the default, okay? This is the lactose operon. What should be the cue to remove the repressor and allow transcription? What should be the environmental cue to remove the repressor and allow transcription of these lactose metabolism genes? If you're not sure, you can put a question mark, but do put something in, just to sort of like force yourself to pay attention. Great. So the environmental cue is going to be lactose. Lactose is what will remove this repressor. Let's see that happen. So here it is in its functional shape. If lactose is around, it binds and turns it into the non-functional shape. It tells it, you know what, dude, you've been working so hard. You need to go take a break. Put your feet up, relax. And then suddenly we have the possibility of transcription happening. That's going to cause all of these lactose metabolism genes to be expressed. Okay, so when lactose is not present, when lactose is absent, the default state for the repressor is what? Active or inactive, functional or non-functional, ready or lazy. Lactose is absent. There's no lactose in the environment. Right, the repressor is born ready. Since it's born ready and it's sitting in the operator, what's happening with transcription? It stopped. Yeah. And again, this is what we wanted, right? If there's no lactose, we do not want to make enzymes to metabolize lactose. That'd just be a waste of energy. Now let's add some lactose. What happens to the repressor? We add some lactose, it binds to that active repressor and makes it right. We add lactose, the repressor goes away. But you might think, oh, the next thing is that transcription happens. It just turns out to be more complicated than that. If you've done the prep, you know a little bit why this is. Maybe you know something and you don't completely understand it, but you know there's more to the story. Into the chat, write anything related to this why. Why am I saying that once the repressor goes away, we don't necessarily get transcription right away? It does rely on something else besides just lactose. Yeah, and it does have to do with the promoter, John. Okay, so let's talk about this. We're not going to take our break yet. We're not quite ready. We've got 10 minutes. Here is a picture of the genes that are part of the lactose operon, or the proteins that are made by that operon. One of them is a permease that brings lactose inside. One of them is an enzyme that breaks lactose in half. What is important to understand now is that lactose, like many, many, many other sugars, is not the best carbon source. What is the best carbon source? What is the best fuel for most cells? It's true of your cells for the most part, and it's true of many microbes. The answer is glucose. The answer is glucose. And it's true that there's some glucose in lactose, but there isn't as much glucose in lactose as there is glucose in glucose. Okay, so I'm going to give you just something. We're not going to go into the details of why glucose is the best, except to just say, remember in metabolism when we talked about glycolysis, the Krebs cycle, and the electron transport chain. Those metabolic pathways are set up for glucose. You can use other sugars if you have the right enzymes to change them into something that's kind of like glucose. either change them directly into glucose or change them into something that is a breakdown product of glucose. But either way, if you have to change them into glucose, that costs energy. And if you have to put them into glycolysis lower than the starting point, then you're going to miss some of the energy generating steps. And it costs energy. So the best molecule, if it's available, is glucose. Now, many organisms, many cells, preferentially consume glucose, especially microbes, preferentially consume glucose if it's available and ignore everything else until the glucose is gone. Now, I'm going to tell you two things. First, I'm going to try to give you a reason why that makes sense, and then I'm going to hopefully help you to understand how we can make that happen. So just imagine two bacterial cells, okay? I'm going to call one Bob and one Stanley, okay? Cell Bob is not a picky eater. He'll just eat whatever passes by. If there's a glucose molecule, he'll eat glucose. If there's lactose, he'll eat the lactose. If there's a glucose and a lactose right next to each other, it's like a 50-50 split which one he reaches for first. He really couldn't care less. When he's eating these sugars, let's just assume the environment both of these cells are in has glucose plus a lot of other things. And so we got Bob over here. He eats a molecule of glucose. He eats a lipid. He eats a xylose. He eats whatever. And when he eats the glucose, he gets a lot of ATP, and everything else that he eats gives him less. Over here, we've got Stanley. Stanley, the cell, is a super picky eater. He will eat everything eventually, but if glucose is around, he eats the glucose first exclusively. So he's sitting right next to Bob. The glucose passes by, he grabs the glucose. Glucose and lactose pass by, he grabs the glucose. Glucose and lipids pass by, he grabs the glucose. Every time he takes the glucose, he gets the most energy. So every time he's in a mixed environment with Bob, Stanley is getting more energy per unit time because he is preferentially using the glucose. More energy means he can go through cell division faster, and all his kids are going to be picky too. Who is going to win the evolutionary race? Bob, who's like, I'll eat whatever, I don't really care, or Stanley, who's like, I'm going to hug all the glucose until it's all gone. Yeah. The cell that preferentially uses glucose when it's available is the one that's going to be more successful, have more progeny, and win if the battle of evolution, which is the case in microbes, is about having more kids. So there's a very strong evolutionary pressure to preferentially use glucose. It's not to say that those cells won't use other things, but they will use the glucose first and outcompete cells that don't. So how do we make this happen? And what do we want then? Like, the lactose operon metabolizes lactose, which is a non-optimal carbon source. It's not glucose. It's not as good. So do we want the decision about making these enzymes to just be about, is lactose present? Or do we want this decision to be about, is lactose present and is glucose present? Do we want to care about whether glucose is around? Yeah, we do. If we're Stanley, we do. So let's think about how we can make that happen. Okay, so I'm just going to skip through this again. We've talked this to death, I think. Okay, maybe I will go through this. So in the absence of lactose, the repressor is active. That's its default state. It binds the operator. It prevents transcription. That's all to the good. There's no lactose. There's no need to make enzymes to break down lactose. Consequently, we don't have any transcription. We add lactose. Now, I want to make sure that this is clear. Lactose binds the repressor and inactivates it, and that's still true. The repressor is sensitive to lactose. The repressor cares about lactose only. Okay, so the repressor is not the one that's going to help us include this glucose readout. The repressor cares about lactose, and if lactose is absent, it's preventing transcription. If lactose is present, the repressor goes away. that opens up the possibility of transcription. However, this is where things start to get more complicated. The type of promoter where RNA polymerase sits down that is associated with inducible operons like the lactose operon and with all other non-optimal carbon source catabolic operons is a shitty promoter. It's not very good. My favorite analogy that I will give you when we're in lab together is to think about RNA polymerase like an airplane that's flying over airports, right? And big airports like SeaTac and LAX are very easily spotted by that airplane. It can land there because it sees the runway is massive, there's lights, there's a control tower. It's very obvious there's a runway there. Those are really good promoters. Some promoters, like the ones associated with inducible operons, are like Farmer Bob's cornfield, okay? They're a place where, while technically speaking, a plane could land, if you were flying over it and looked down, you would never know that. The lights are off, there's no control tower, there's no flags, there's no indication that this is an airport. So these types of promoters actually need an additional activator in order to make the promoter visible to RNA polymerase. And activators are the opposite of repressors. Activators exert positive control instead of negative control. But like repressors, they bind to DNA and they're allosterically regulated, which means they have two possible shapes, one that binds the DNA and one that does not. The one that we're going to talk about is CAP. CAP stands for catabolite activator protein. It also stands for cyclic AMP activated protein, and it is sensitive to cyclic AMP. It is able to bind the region next to these crappy promoters of catabolic operons and helps them to be sensitive to glucose. So I want to say a few more things about that, and then we're going to go through some animations. And let's check my time. Okay. I want to say something about CAP, and then I think we'll do the animations after the break. And if you're sitting here sort of going, oh my God, I have no idea what she's talking about, this is really complicated, we're going to go through these pictures. It's going to make more sense. So I'm just begging you to please hang in there, not give up on me. Don't give up on yourselves, but just take in as much as you can. And then we're going to do a lot of pictures. CAP is sensitive to cyclic AMP. So something you need to know about this is that cyclic AMP is actually a really good measure of glucose levels. And the reason for that is that ATP and cyclic AMP are both derived from the same original molecule. So they are inversely related to each other. When there is a lot of ATP, it's using up the starting molecule and cyclic AMP goes down. When there's not very much ATP, then that starting molecule is not being used up and cyclic AMP is high. And what condition makes ATP go up the most is glucose. If there's a lot of glucose, there is a lot of ATP. And what do you think happens then to the levels of cyclic AMP? High ATP, low cyclic AMP. Since CAP is sensitive to cyclic AMP, if there isn't very much of it, it is not functional. So CAP is a good readout of glucose. If glucose goes away, ATP levels drop. What happens to cyclic AMP levels, do you think? They go up. And when they go up, what happens to CAP, which is dependent on cyclic AMP? When cyclic AMP levels go up, CAP can become activated. And then it goes to bind these operons for catabolic enzymes, not glucose, and it is an activator. So how how does this particular activator work? I'm going to give you an analogy for this too. Think of CAP as like the lights that come on at the airport. RNA polymerase is flying over Farmer Bob's cornfield, but CAP suddenly sits down, turns the lights on, and the RNA polymerase is like, holy crap, there is a runway there. I had no idea. So CAP makes it possible for RNA polymerase to find that promoter. Okay, so we're going to go through this and through this and through this in a little bit after the break with some pictures. But for right now, let's take 10 minutes to just rest and relax. So 6:02, I will see you again. 6:02. Welcome back, everyone. Okay, now it begins the pictures. And I hope that this helps you to remember this better. It gives you a template for how you should draw your own pictures when studying and what you should have in your study guide and available to you during an exam. Okay, so remember that the lactose operon is more complicated than the tryptophan operon because there's not just the repressor we have to worry about, which is going to exert negative control and prevent transcription. There's an activator we have to worry about, which is going to encourage transcription. And all of this comes about because there is a need to pay attention to two things, not just the metabolite itself, which is lactose, but also a other environmental signal of glucose. And there's a couple things that are needed to achieve this kind of regulation. First of all, we need a crappy promoter that is going to require the lights to be turned on in order for RNA polymerase to bind. And we're also going to need a repressor that is by default active. So when we look at this, right, the negative regulation, that is to say the thing that's going to prevent transcription, is exerted primarily by the repressor. It binds the operator, and that happens if lactose is not around. Right, so here's the repressor. It's bound to the operator. This is its default state. When it's made, it's already in this shape that it can bind the operator. The positive regulation is exerted by CAP. The thing that binds the CAP binding site is CAP. And this binds the CAP binding site if glucose is not around. And this is necessary in order to bring RNA polymerase to the promoter, because again, this is a bad promoter. And the only way that RNA polymerase is ever going to find it is if CAP is there telling RNA polymerase, hey, yo, there's a promoter here. Did you know that? Here's a promoter. RNA polymerase is going to be like, I never saw that before. So one thing to know is that the repressor binds the operator if there's no lactose, and CAP binds the CAP binding site if there's no glucose. And both of these can be removed if the sugar to which they are sensitive is present. Okay, so what controls CAP? Before the break, we talked about this. What controls CAP directly? It is sensitive to or activated by what small molecule? Glucose indirectly, yes, but most directly, yes, Sarah. Cyclic AMP. CAP is controlled by cyclic AMP. Cyclic AMP binds and activates CAP. If cyclic AMP levels are low, CAP is not active, so it's not going to bind the CAP binding site. Cyclic AMP levels are high, CAP becomes activated and binds the CAP binding site. So when glucose is high, which means ATP levels are high, which means cyclic AMP levels will be low, is CAP active or not? Correct. If cyclic AMP levels are low, CAP is not active, which is to say, if glucose is high, CAP is not active. Okay, and it's not because glucose is binding CAP directly, it's because cyclic AMP is required to activate CAP and cyclic AMP is not around. If CAP is not at the CAP binding site, the lights are off at the airport and RNA polymerase cannot find the promoter, and so there will not be any transcription. What happens when glucose levels drop? ATP levels drop, cyclic AMP levels rise. CAP is now right, CAP becomes activated. Cyclic AMP binds to it. CAP becomes activated. It binds to the CAP binding site. The lights come on at the airport, and RNA polymerase does what? Binds to the promoter. Okay, so let's have a look at what's happening here. Okay, remember our ultimate goal is here are the lactose metabolism enzymes. We want these expressed only when lactose is around and glucose is not around. So this is sort of the situation where if there's no sugar, right, remember that the repressor is born ready. And so it's sitting in the operator, okay? Let's say there's no glucose, there's no lactose. Then the repressor is here because there's nothing to remove it. There's no lactose. And because there's no glucose, ATP levels are low, cyclic AMP levels are high, CAP is active, it's bound to the CAP binding site, the lights are on at the airport, RNA polymerase is here. Are we getting transcription? Well, it looks like we could, but the repressor's in the way. All right, yes, the RNA polymerase is there, but the repressor's in the way. So are we getting transcription? Let's wait for everyone to weigh in here. I have 2 answers so far. We have 15 people on the call, and I'll wait for a few more answers. If you're not sure at all, question marks are always welcome. In this circumstance, there's no lactose, so the repressor is on the operator. There's no glucose, so CAP is here, and it's brought RNA polymerase here. So it's just like this scenario you see right here, where yes, RNA polymerase is there, but repressor is in its way. Transcription is not happening. Right. Okay. Now, what if there's only glucose, still no lactose? Okay, still no lactose. So the default situation for the repressor is that it's bound to the operator. There's no lactose, so the repressor, is it still here or is it gone? No lactose, is it still here or is it gone? Remember that the repressor is only removed when lactose is around. Yeah, still here. No lactose, repressor still here. Okay, but we've added glucose. ATP levels went up. Cyclic AMP levels went into the toilet. CAP, is it bound here or not? Is CAP active or not active? Correct, CAP is gone. Glucose is around, ATP is high, cyclic AMP is low, CAP is not here. So is RNA polymerase here? The lights got turned off at the airport. Can RNA polymerase land the plane? That's right, it cannot. Are we getting transcription? That's right, we are not getting transcription. Before we move on, let's just ask ourselves if this is all what we wanted, right? Do we want transcription when glucose is around but no lactose? Do we want to be making enzymes to metabolize lactose if there's no lactose? That's right, we don't. Okay. So in both these scenarios, we have what we want, which is we're not making these enzymes. We're not wasting energy making those enzymes. Okay, great. So now let's imagine a world where we still have glucose, but we add in some lactose. Before we talk about what's happening here, let's talk about what we're hoping to achieve, okay? Suddenly, yes, there's lactose, okay? And there's glucose. Remember Stanley and Bob? Who was winning? Stanley was winning. How did he win? Did he just have a little bit of column A, a little bit of column B, little glucose, little lactose, little sugar in my life? Or did he preferentially choose to eat up all the glucose, hoping to hog it for himself? Yeah, he was trying to hog all the glucose. So both sugars are present, but we really only want to be metabolizing glucose. Do we want to be expressing these genes? Even though lactose is present, do we want to make the metabolism enzymes? Is that like ideal for us? Remember, if both sugars are present, what we want is to only eat the glucose. Ignore the lactose until the glucose is all gone. Do we want to make lactose metabolism enzymes if our goal is to ignore the lactose? That's right, we do not. I know I really had to talk you into that. You were like, but I still kind of want the lactose. No, you don't. All right. So let's see if we can make that happen. First, let's talk about the lactose. Because it's present, remember when lactose binds the repressor, it convinces it to go take a break. So now the repressor is gone. Bye-bye. Uh-oh, we don't want these to be made, and yet here we are halfway to transcription. Let's see what's going on with glucose. Glucose is there. ATP levels are high or low. Glucose is present. ATP levels are high. Okay. Cyclic AMP levels, therefore, are low. CAP, active or not? CAP is not active. Okay, so it's not bound to the CAP binding site. So the lights are off at the airport. This is a shitty ass promoter. Is RNA polymerase here or no? Did RNA polymerase land the plane with the lights turned off in this bad airport? It did not. Okay, so we don't have RNA polymerase. Phew. Okay, so even though the repressor is gone, we don't actually have any RNA polymerase. So did we achieve our goal? We managed to avoid making these enzymes prematurely. Yeah, good. CAP's not active, RNA polymerase therefore cannot bind, transcription is still not happening. Okay, what are the conditions that we want to be when and only when we will turn these enzymes on? When do we want to actually transcribe these enzymes for metabolizing lactose? What is the environmental situation? Lactose present and glucose is absent. No glucose. We want lactose only. That's what we want. Okay, so let's talk about lactose only. First, here's the default repressor, but in the presence of lactose, what happens to this repressor? Yeah, lactose binds, convinces it to take a coffee break, little creamer in my coffee, and off it goes. There, it's going away, bye-bye. Meanwhile, no glucose. No glucose means ATP levels are low, cyclic AMP levels are high, CAP is activated, it brings RNA polymerase to the promoter. Do we finally get transcription? Yes, we do. Thank goodness. Okay. Now I want to tell you something that I hope does not harm you too much. This is not the only place in the genome where this is happening. The repressor that we've been talking about, this one, that binds the operator or doesn't bind the operator, depending on the environment, is sensitive to lactose. This repressor is unique and specific to this operon. This is the lactose repressor. This is the lactose operon. This is the lactose operator. It only binds here and regulates this operon. But CAP is different. CAP is a universal. CAP binding sites are not specific, right? This CAP binding site is here in the lactose operon, but the exact same CAP binding site is also in the xylose operon, and the arabinose operon, and the mannitol operon, and the mannose operon, and the erythritol operon, and all of the other oses and atols, all the other sugars that are just not as good as glucose, and they're all inducible. So when you think about what's happening, I'd like you, if possible, to think about it a little bit more globally, that the whole genome is affected by the environment. It's not just this one little area. The reason that this is so elegant and so efficient is that the repressor for each operon can help us to make sure that we're not transcribing stuff we don't need, because it's going to be sensitive to that presence or absence of that thing. But CAP is going to come in and say, but you know what, glucose is around, so, or glucose is missing, right? What CAP is a little bit like, I don't know if you ever have these people at your workplaces are kind of like the safety marshal or something if there's a fire drill. CAP is kind of like somebody who, if there's an emergency, and the lack of glucose is kind of a cellular emergency because suddenly ATP levels are low and the cell maybe could die. CAP is alerted by that situation. Basically, cyclic AMP comes and says to it, hey, guess what? We ran out of glucose. And CAP is like, holy shit. And it runs all over the place in the genome and sits down at all these CAP binding sites and says, hey, RNA polymerase, you know these promoters you've been ignoring. Come on, sit down here and here and here and here. And basically, it doesn't know whether or not this particular lactose operon is going to be used or not. It doesn't know if lactose is present. It doesn't know if xylose is present, but it's just sitting down in case. And this repressor is going to go away if that carbon source is available, and it's not if it's not. And so then we don't waste energy making stuff we don't need, but we're ready in case lactose or something else comes along. Does that make sense? Yes, no, or kind of. Again, pictures, pictures, pictures. And one of the reasons that I bring up the existence of other operons is that this week in lab, for our transformation reaction, we will discuss the arabinose operon. And guess what? Arabinose is also a shitty carbon source, just like lactose. And if there was arabinose plus glucose, which do you think the cell should use first? Yeah, glucose. And so the arabinose operon is organized just like this. There's a CAP binding site. There's an operator, though it is specific to the arabinose operon and the arabinose repressor. And there are genes that are specific to arabinose metabolism. And that's all I'll say today, but when we get into lab tomorrow, you'll see a picture just like this, but for the arabinose operon. And we're going to talk our way through this again, but with arabinose. Okay, so now all of this brings me to the phenomenon known as catabolite repression, which is a fancy word of saying picky eater. Okay, so let me tell you first the fascinating data that got people to realize this was happening, because just like you probably walked in today not knowing that this is a thing, people who were originally doing studies of growth in bacteria had no idea either. So on the left, what we have is two growth curves. It's the same species, but they're just grown in different sugars. So here is the broth. This is the growth curve of a bacterial species X grown in glucose only. And you can see it's in log phase right now, so its numbers are going up pretty quick. Over here is the growth curve of the same exact species grown in a broth with just lactose. And what do you notice? Which sugar did they grow faster in? Yeah, they obviously grow faster in glucose. And remember, I told you that glucose is a better carbon source. Per glucose, you get more ATP. Per unit time, you get more ATP. That means you can divide faster. That means your population is going to grow faster. Your generation time is going to be shorter. Now, if, as people assumed initially, if cells were not picky, and let's say they were all Bob, and they're now, let's put them in a broth that has both glucose and lactose, and whatever molecule drifts across their field of reference at that particular minute time point, they'll eat it. Glucose drifts by, they eat it. Lactose drifts by, they eat it. And they don't care particularly which happens in what order. What would you predict the growth curve would look like? Would it look like this, the glucose curve? Would it look like the lactose curve? Would it look like something in between? If they were not picky. They were not picky. So that means, like, you know, let's suppose you get 10 ATPs per second eating glucose, and you get 5 ATPs per second eating lactose. If you're eating a glucose, then a lactose, then a glucose, then a lactose, on average, you might expect you'd be getting like 7 ATPs per glucose per second, right? So where would your growth curve be? Like, would it be glucose, lactose, in between, below, above? Where would it be? In between. Yeah, I agree. The prediction is that you would see something like this. But that's not what they saw. They took this species that was doing better in glucose than in lactose, that they would have predicted if both sugars were present would just kind of give you an intermediate result. And they put it into that broth with both sugars. And instead of seeing this straight line, they saw this staggered line. Okay. The first part of this growth curve looks like the glucose growth curve. Then there's a pause. And then the second part looks like the lactose growth curve. So what happened right here at this inflection point? Yeah, it's adjusting, but yes, Sarah, yes, Haley, it ran out of glucose. And then now it's in basically an adjustment period, we'll call it, because I have a question about this and I don't want to give it away, adjustment period. And then now what is it eating? It is eating that. It is eating lactose. Great. Okay, so we're not taking our break now. Instead, we're going to answer some questions. So we're going to hearken back to what we remember about the growth curve, different parts of the growth curve in the growth section. Here is a growth curve of that cell species grown in glucose plus lactose. It's E. coli. We see that there initially you put the cells into the broth and there's no change in cell number because they're doing their adjustment period. Then they take off, grow really fast, eating up the glucose. Then there's an adjustment period, and then they take off more slowly, eating up the lactose. Which of these best describes the phases or of growth that they're in these different time points? Is it lag, log, stationary log? Is it lag, log, or is it stationary log, stationary log? A, B, or C? What do you think? Great. So we have guesses all over the spectrum. Okay. Let's talk about it. So we put the cells in this fresh broth and there's lots of food, but it's a new environment. They have to adjust, make new enzymes. What do we call that? Yeah, that's lag phase. So it's going to be one of these two. Then they take off, they're growing really, really fast. Obviously that's log phase. Boom, they run out of glucose. But did they run out of everything? They did not. They're now in a lactose world. So they've got to make new enzymes, right? They've got to make the enzymes to metabolize lactose. So what is this? Yeah, this is lag phase. Stationary phase would only happen if they ran entirely out of food. But what's happened here is really they ran out of one of the carbon sources. The other one is untouched. So they're just in a new environment, basically. They ate their way to a new environment. And then they take off, okay? If there was another flat line here, what would it be? Stationary phase, right? Because then they would have run out of both sugars. Great. Let's talk about this. Okay, so A is referring to the steep curve and B is referring to the shallow curve, or line, I should say. Which of the following is correct for the time marked A, which is the steep log phase on the graph? Again, what this is E. coli grown in both glucose and lactose together. So just read the options. Tell me in the chat which you think A, what's accurate for A, thinking about what's available in the environment and what's happening at the genetic level. Take a second, think about it, put your answer in. If you can't get there, put a question mark. I need everybody who's got like brain cells and hands. So if you're driving, don't do this. Or like carrying a patient at work, but everybody else needs to answer this question. All right, we've got 4 answers so far. Let's see if we can get some more in. We've got 5 answers. We have 14 people on the call. I'll wait for a few more answers to roll in. Okay, let's talk about it. Before I reveal the answer, let's talk about it. So in A, we have both sugars available in the environment. It is really important that you get used to solving these logic problems by taking one aspect at a time. Let's start with the lactose. Because lactose is present in the environment, it has bound to the repressor and inactivated it. So is the repressor bound to the operon or not bound to the operon in its inactivated state? Right, lactose being present has inactivated the repressor, so it's not bound. So that narrows it down to A and B. So we can get rid of these other 2 options. Because glucose is present, ATP levels are high or low? Yeah, they're high. Consequently, cyclic AMP levels, which are the opposite, are going to be low. Is CAP active or not active? Not active. So CAP not active, that means it's not bound to the binding site. Okay, that narrows it down to A, not bound to the binding site. And then the enzyme beta-galactosidase is one of the enzymes that is is part of the operon. So it's not being transcribed. Does that make sense? Yes? No? Kinda? Great. Let's move on to the next part right here, B. Now in B, what's happened, of course, is that we have run out of glucose. What's left? Lactose. Okay, with that in mind, lactose is present in the environment, glucose is not present. Which of these best describes what's happening at the genetic level? Take your time, put your answer into the chat, and we'll see how we do. Great. Okay, so let's talk about it. Lactose is present, so it has disabled the repressor. So that narrows it down to A or B. Okay. Glucose, however, is gone. Glucose is gone. ATP levels are low. So cyclic AMP levels are going to be they're going to be high. CAP is going to be activated. And that means it's going to bind to the operon. That turns the lights on at the airport. RNA polymerase is going to sit down. The repressor is not in the way. Are we going to get transcription? Yes. And that is what's happening. And that's what we want too, because this enzyme is necessary to metabolize lactose, and lactose is the only carbon source available. So we have achieved our goal. Now, imagine that we have reached a stationary phase, which would be the flat, like if once we run out of both glucose and lactose, we're finally in stationary phase, because those were the original two sugars and now they're gone. So we really have run out of food, like for real. What happens in stationary phase? Both sugars are gone. Put your answer into the chat. All right, let's talk about it. Let's start with the fact that lactose is gone. Lactose is gone. So where's the repressor? Remember, this is the repressor that's born lazy, not born lazy, born ready. Repressor's born ready, doesn't need lactose to tell it what to do. So it's sitting in the operator because lactose is gone. It's bound to the operator. Okay. There's no glucose. No glucose. It's an emergency. Where's CAP? Cap is active, cap is bound. Okay, now they're both bound. But in that case where they're both bound, yeah, RNA polymerase is there, right? But so is the repressor. Are we getting transcription? Are we getting transcription, guys? RNA polymerase is there, but the repressor is in the way. Are we getting transcription, yes or no? That's right, we are not. Okay, so what is the answer? What's the answer? Yes, sometimes the answer is none of the above. And you're only going to know that if you pay attention to detail. Both of these have aspects of them that are correct, right? They both say the repressor is bound, which is true. This one says cap is bound, which is true. This one says the transcription's off, which is true. But there's an incorrect part of these statements. It is E. Cap is bound due to no glucose. Repressor is bound due to lack of lactose, no transcription. Yes, that was tricky. That was on purpose. Okay, pay attention to detail. Okay, let's talk about this one. Let's suppose that E. coli developed a mutation in the gene for the lactose repressor. It changed it so that that allosteric site doesn't work anymore. Lactose can be around all day. The repressor does not change into the inactive state, right? Can't bind lactose. What's the consequence? Remember, it's always fine if you have no idea to put question mark, but it's more fun to guess even if you're wrong, in my opinion. Great. So what would happen if the lactose repressor could not bind lactose? It's born ready, it sits in the operator, but without the ability to bind lactose, it can never be removed. Since it could never be removed, can you ever get transcription of beta-galactosidase? So what's the answer? Right. Okay. Let's do a little fill in the blank fun times. Lactose operon is one that breaks down lactose, making it catabolic or anabolic. Yeah, it's catabolic. Catabolic operons for things that are not glucose are typically regulated as inducible or as repressible. If you don't know, you can put a question mark, but if you do know, put in your answer. What was the lactose operon, right? Is it inducible or repressible? Remember with our definitions. Inducible means the default is no transcription and then the environment can turn it on. Repressible means the default is yes transcription, the environment can turn it off. Yeah. This catabolic operon is typical in that it is inducible. Environmental signal lactose therefore acts as an inducer, correct. By default, is the repressor born ready or born lazy? Is it born active or inactive? Is it born functional or non-functional? Yeah. It's born ready. It's born active. By default, therefore, transcription is on or off. Correct. It's off. Let's look at the tryptophan operon. It synthesizes tryptophan, an amino acid. Therefore, is it anabolic or catabolic? Anabolic. That's right. We want by default for it to be on, but we want to turn it off sometimes. Is that repressible or inducible? The environmental signal tryptophan acts as a co-repressor. That's right. Because we want our default state to be what it is, the repressor, is it born ready or born lazy? Active or inactive? Functional or non-functional? Correct. The repressor is born in the inactive, lazy, non-functional conformation. Transcription, therefore, is on by default. Okay, last few minutes, we have something to talk about, which is how genetic change can occur. All organisms that are DNA-based, right, and even viruses that are not necessarily DNA-based, anything that replicates using nucleic acids changes over evolutionary time due to mutations, which are introduced during the process of cell division due to errors in the copying of the genetic material. That's true of you, that's true of microbes. However, what's different in the microbial world, what's different in the prokaryotic world, because this does not really happen so much in eukaryotes, what's different in the prokaryotic world is that in addition to inheriting DNA from their parents, which is where the mutations come from for us, they can acquire DNA from their peers. This would be like if I went to coffee with a friend, and when I left, I had her some of her genes, right? That doesn't happen to people. Happens to microbes, and here are the three major mechanisms: transduction, conjugation, transformation. All of them are so-called horizontal gene transfer events as opposed to vertical transmission of DNA from parent to child. So horizontal is sort of peer-to-peer. All right, transformation is defined as the naked DNA uptake by bacteria. Bacteria are walking around minding their own business and they spot a DNA molecule laying on the ground and they take it up. Where did that DNA molecule come from? It came from a dead cell. Conjugation is the equivalent of bacterial sex. Two living cells physically connect and exchange DNA. And transduction is when bacterial DNA is transferred by a viral intermediate. This is a very famous experiment. And what happened showed one of these mechanisms at work that sort of jump-started this whole field of inquiry. But this is Streptococcus pneumoniae, an encapsulated streptococcus. The capsule is one of its virulence factors. It causes pneumonia in people, and when you inject into the mouse, it causes a lethal disease. This bacteria replicates in the bloodstream. You can isolate it, look at it under a microscope, and see this same exact organism. There's a sibling strain with a mutation in the capsule-making gene. It means it doesn't make capsules, doesn't cause disease in people, doesn't cause disease in mice, can replicate. We can re-isolate it. Look under a microscope, it looks like this. We know it's the harmless version. We can take the bad kind and heat kill it and inject the mice. The mice stay healthy. It's dead, so it doesn't replicate. But when we take the heat killed and the harmless one together, the mouse dies and we recapitulate the original. What mechanism is this? Yeah, this is transformation. Raise your hand if you can explain what happened. Why did these living cells turn into basically these other dead cells? It's not that these guys reanimated. What happened? All right. What I'm going to do is I am going to leave that for now. I'm going to warn you that I'm going to cold call you on some stuff about this last part of the curriculum, which is the conjugation, transduction, transformation stuff on Monday. And we will start here. These are just a few slides at the end. We'll start here, do a little cold calling at the beginning of class. We're going to do a Kahoot, and then we'll launch into viruses. So one thing that's really important to say at this point, let me see if I can go back, is that you need to prep for the weekend, okay? So what is going to happen is there is a genetics review quiz. There is a deadline for lab, but you need to prep for Monday by doing the viruses video slides. This is a Tuesday deadline, sorry. But you need to prep for Monday by doing the viruses curriculum. And there is a worksheet for that that you'll want at least by Tuesday. So make sure to do your prep, watch those video lectures, don't skimp. It is vitally important that you watch them. And come on Monday also prepared to cover the last few slides in this slide deck. Meanwhile, I'm going to pause or stop the recording and take any questions that you have.